All right, so now we're going to talk about carbohydrate structure, okay? And remember that carbohydrates are the largest fuel molecule in the human body. They are composed of monomers of simple sugars, so they're composed of monosaccharides. You can get disaccharides and polysaccharides, okay? Di means two, poly means many, all right? So monosaccharides are the simplest. And then the molecules themselves exist in two different primary structures, either aldoses or ketoses. So here is the structure of D-glyceraldehyde, which is an aldose. And it's called an aldose because there's an aldehyde moiety here. Okay. And then we have dihydroxyacetone, which is a ketose because there is a ketone moiety here, right? The other component to this is the fact that they have some kind of direction, right? And so let's look at glyceraldehyde more carefully down here, where you can see that in the Fischer projections, we can have a right-handed or a left-handed glyceraldehyde molecule because there is a steric carbon here in the center, right? So the location of where the OH is determines whether it is right-handed, D, or left-handed, L. And D-glyceraldehyde, okay, means that the OH is on the right-hand side of the Fischer projection, and L means that the OH is on the left-hand side of the Fischer projection. And I will tell you that the sugars that we metabolize in nature are always the D form. Okay, so there is always direction, and the way in which these are going to form the polymers is going to become important in a moment. Okay, all right. So we have now just a simple a diagram of just the way in which we start counting carbons and we start naming these molecules, okay? So the base structure, as I said, is D-glyceraldehyde. Again, the OH is on the right-hand side, and it has an aldehyde moiety at the top. This is hard to see, I know, in the recorded lecture, so just pull open your book and you can take a look at it. 3 carbons, 4 carbons, 5 carbons, 6 carbons. Almost all of the sugars that we metabolize for fuel are 6-carbon molecules, right? And the two most important are glucose and galactose, okay, of the 6-carbon sugars that are aldoses, okay? Now we have our ketoses, right? And again, the base structure here is dihydroxyacetone, 3 carbons. Then we have 4, 5, and 6 carbons, and the most important one is D-fructose, okay? You need to know the structures of glucose, galactose, and fructose. All right, and the first extra credit question here is, the most common sugars metabolized by the body are in the L configuration. True or false? All right, so now let's talk a little bit about the chemistry of these molecules, okay? We know from organic chemistry that an aldehyde and an alcohol will react to form a hemiacetal. Okay, so here's our aldehyde and here's our alcohol, and you can see that we'll form this hemiacetal, which is this structure right here. All right. We also know that a ketone or an alcohol will react to form a hemiketal. All right, those are reactions that you should remember from organic. The uniqueness or reason why this relates to sugars is that sugars have in them an aldehyde and an alcohol, in this case D-glucose, and those two entities can react to form the hemiacetal. And the hemiacetal simply cyclizes the glucose molecule, right? And we get a structure that resembles either puran, if it's an aldose, or furan, if it's a ketose, all right? And so you can see here with glucose that we create a new steric carbon here. And so we have to have an additional designation, either alpha or beta, to indicate where in the molecule the OH on this new carbon, this new stereocarbon, ends up. So here we have alpha D-glucose. The alpha means that the OH is below the plane of the molecule. And if it were beta D-glucose, the OH is above the plane of the molecule. All right. The same cyclization happens for fructose, only now we make a hemiketal, right? Because again, we have the ketone moiety, and here we have an OH. These react, okay, to form the ring structure, and now we have a 5-membered ring, but 6 carbons, okay? Whereas the glucose molecule is a 6-membered ring. okay, a 6-membered ring, right? So both molecules have 6 carbons, but the hemiacetal is 6 members of the ring, and the hemiketal has 5. All right, and just as with glucose, we have an alpha D and a beta D fructose, okay, depending on where the OH is. Again, below the plane is alpha, above the plane is beta. All right. The other sort of structural component here that's important is to recognize that the most stable conformation of that ring structure that we have for glucose, so if we look at our glucose ring structure, is not a planar molecule. This is not a very stable structure, this Fischer projection. In fact, glucose exists, if you'll remember, in 2 separate forms, either the chair form, and it's a chair form because it resembles a chair, or the boat form, okay, because it resembles a boat, right? The most stable form of glucose is the chair form. And this is because it aligns the OHs in the most sterically favorable location in the molecule, and so thermodynamically it's the most stable, right? So that brings us to question 2, which is the most stable form of glucose is the blank form. All right, so now we're going to talk a little bit about some other things we can do. So these structures here demonstrate the advantage of having OHs on a molecule, right? We can add substituents to the glucose molecule, and we can create a very diverse collection of sugar moieties. These are very important later when we talk a little bit about the other things sugar does besides being a fuel molecule, right? So here we've added something. This molecule is called GalNAc, okay, because it has this acetylgalactosamine. This molecule is called acetylgalactosamine because it has an amine and an acetyl group on it, and it's attached to a galactose molecule. All right. Here's GlicNAc, okay, which is just a glucose base that has the same amine and the same acetyl group over here. Okay, these molecules ultimately form big, giant, large structures that exist on the outside surfaces of cells in the membrane to sort of identify them. So you're going to see that these become tags for cells. Okay, and we find that they're either O-linked or N-linked glycosidic bonds. All right, if it's an N-linked bond, it's because it has an amine. And if it's an O-linked bond, it's an O bond or an ester bond. Okay, and we're going to talk about those a little bit later. All right. All right, so the other thing we've sort of mentioned or alluded to before when we were talking about the role of phosphates or some of the common themes of metabolism is that we can also add things to these OHs. So again, remember that the functional group that's on these sugar molecules are these reactive OHs. And here is glucose 6-phosphate, and what we've added here is a phosphate molecule. Okay, we've made a phosphoester bond. Okay, here's the same thing. We've added a phosphate to dihydroxyacetone. And here's glyceraldehyde with a phosphate on it, okay? These are 3 very common molecules that are intermediates in glycolysis. And again, what we said the phosphate does is it primes these compounds for reaction. All right, so now let's talk a little bit about the glycosidic bond, okay? Because this is incredibly important. The glycosidic bond is a bond that forms between 2 sugar molecules. The reaction here is a condensation reaction. Remember, all reactions that form the macromolecules are condensation. So it's the loss of a water molecule, or dehydration synthesis. Okay? And what becomes very important is the direction with which this bond forms. And so remember, direction is very critical in macromolecules. So this illustrates an alpha 1,4 glycosidic bond. And it's an alpha 1,4 because it forms between carbon 1 of the first sugar, which is in an alpha position, okay, which means the OH is below the plane, and then it forms with the 4th carbon of the next sugar in line. And that's why it's called an alpha 1,4 glycosidic bond. All right. We have lots of different, okay, glycosidic bonds. So here is an alpha 1,2. Okay, again, it's alpha because it's with the OH is below the plane. it's between carbon 1 of a glucose molecule and carbon 2 of a fructose molecule. Okay, so this is an alpha 1,2 glycosidic bond, right? Here is a beta 1,4 glycosidic bond. It's beta because the OH is above the plane. It's between carbon 1 of a galactose molecule, in this case this molecule is galactose, and carbon 4 of a glucose molecule. This is lactose, and this is a beta 1,4, and lactose is composed of galactose plus glucose. This molecule is sucrose, which is just a glucose and a fructose molecule. Okay, so let's not forget what this is. This is sucrose. All right, so sucrose is an alpha 1,2 glycosidic linkage between a glucose molecule and a fructose molecule, right? And now we have maltose down here, which is an alpha 1,4 between two glucose molecules, all right? And that becomes very important, which you can't see that, sorry. All right? Okay, we can also make, these are chains, so if we were to, these are examples here, by the way, of disaccharides, okay? If we were to continue to add another unit here, okay, to make the polysaccharide, these would be chain molecules, okay? If, however, we want to make a branch, okay, we can also make an alpha 1,6 glycosidic bond, and that's here, an alpha 1,6. So here we have a very small piece of glycogen. These little squiggly lines right here means that the molecule goes on, okay, and that there's more residues as we go further. Okay, same with this little squiggle here. All right, glycogen is a giant polymer of many glucose molecules, and those molecules are attached in a chain with alpha 1,4 linkages, and branches are alpha 1,6. Okay, so now this branch glucose is forming a bond between its carbon 1 with its OH in the alpha position and carbon 6 of this glucose. All right, so now let's talk a little bit about the different kinds of molecules that exist, okay? So the polymers, or the polysaccharides, okay, are the most common carbohydrate macromolecules there are. This bottom structure here is an example of starch and glycogen, and this is just the singular linear structure, not the branch structure. Okay? Starch and glycogen contain alpha 1,4 linkages, and then remember glycogen also has the alpha 1,6 linkages when it's branched, okay? And so does starch. All right, so the branches are alpha 1,6. So starch and glycogen have alpha 1,4 and alpha 1,6. Okay? These molecules are fuel molecules for animals. All right? Animals possess enzymes that can cleave both alpha 1,4 linkages and alpha 1,6 linkages. Okay? Cellulose, which is the plant form of the polysaccharide, is composed of beta 1,4 linkages. Okay? Cellulose is a structural molecule only. Right? We can't metabolize cellulose because we don't have an enzyme that will cleave beta 1,4 linkages. And so we cannot free each of these glucose molecules to use as fuel. Okay? And so you have to remember that only alpha 1,4 and alpha 1,6 linkages are able to be metabolized by animals so that we can free glucoses. Okay? So that brings us to question 3.